In the last presentation, we briefly introduced 5 important themes in biology. In fact, these themes are so important to how we study biological systems that we refer to them as unifying themes. Let's start by looking at how some of these biological systems are organized. Unlike some systems studied by other scientists that tend to reduce their organization over time, biological systems, by and large, are very organized. In fact, regardless of how closely or distantly we look at a biological system, we tend to see such organization. For example, at the tiniest level, we see that lone atoms join together into molecules. Molecules are grouped together and arranged in particular ways to form organelles. Organelles to form cells, cells to form tissues, which form organs and organisms. Even above the level of the organism, however, biologists study genetically related groups called families and populations of unrelated individuals. Often, we mix together different species. We get communities, which integrate with the non-biological world to form ecosystems, which ultimately exchange material and interact with each other in the biosphere. Now, as we go up through these higher levels of organization, things that are more complex can do things that simpler structures cannot. For example, elemental sodium and chlorine are individually explosive, but when they're joined together in the form of salt, they have a unique and delicious taste. That taste and lack of explosiveness is known as an emergent property. At a higher level, organelles such as mitochondria, Golgi apparatus, chloroplasts, and ribosomes, which individually would never be considered alive, are arranged together in a cell, which is very much alive. Thus, life is an emergent property at this level. Up higher still, a population of flowers are all similar to one another and don't have a great way to disperse into new territory. However, the community interaction of bees and other insects with those flowers allows this to happen. There's also a very strong relationship between biological structures and what they do or how they function. A good example of this that we'll look at later in the course is a cell membrane, which surrounds most parts of a cell and plays a very important role in determining what goes into and out of the cell. Due to the structure of the membrane, some molecules pass through very easily and others almost never do. Cells modify their membrane by adding proteins that serve as channels through which larger molecules can pass. So in this case, you can see that the detailed structure of the membrane dictates its function. Now, we know a lot about membranes. Biologists can study a membrane and make inferences about how the cell operates. For example, if you notice a lot of aquaporins or water channels in a membrane, you might infer that moving water into or out of the cell is important for this particular cell, as might be the case for cells lining the large intestine. Robert Hooke saw cork cells for the first time in 1665 using a very basic microscope. He called them cells because they resembled the orderly cells of a monastery. By the mid-1800s, cell theory, the idea that all living things consist of cells, was quite well established. Although it was known that life consisted of cells, the origin of cells was not settled. It took an ingenious experiment by Louis Pasteur to convince skeptics that life could not arise spontaneously. Pasteur had noticed that nutrient broth, if left to sit exposed to the open air, would turn cloudy and be infested with microorganisms, even though none could be seen entering the broth. Pasteur tested the two hypotheses that cells can arise spontaneously on the one hand, and that they cannot and must arise from other existing cells on the other. He boiled nutrient broth to kill the microorganisms in the flask on the top here. The crooked neck of the flask gathered condensate after boiling and clogged up the neck of the flask, preventing anything from moving from the outside into the flask. Nothing grew in that flask. In the lower flask, Pasteur broke the neck after boiling. That flask was therefore open to the air, even though it didn't have anything in it originally, it was open to the air, and microorganisms started growing after a few days. So Pasteur's experiment showed that microorganisms were able to get into the flask from the air, and this inoculation was necessary in order for microbes to grow in the flask, definitively disproving the hypothesis that life could arise spontaneously. The second important theme is that all organisms contain a form of information storage. In all living organisms that we know about, information is stored as DNA. DNA serves as an analogous role as a library. The information is there, but it's a bit difficult to get at. However, the important thing is that it's safe, especially in eukaryotic organisms which have their DNA protected inside a nucleus. However, the DNA isn't very useful by itself, and so the information must get into a form that's more useful. The information is written onto a similar molecule called RNA. You can think of the information being copied from a book in the library, photocopied onto a piece of paper. The book stays protected, and the copy is more likely to be lost or damaged, but the information is reliably copied onto it. Once out of the library, the information on the paper is used to build something useful, in our case, protein. Proteins are used as building blocks of the cell and are really very varied. Their information comes from the complex information encoded ultimately in the DNA. Notice that the flow of information only goes in one direction. Although we now know of some exceptions to this, it generally holds that information flows from DNA to protein, and not the other way around. This flow of information is known as the central dogma of molecular biology, and we'll cover it later in the course. Now, in order to live, all organisms must process energy and transform energy and matter. Almost all energy that enters biological systems originates at the sun and passes through plants and bacteria. These organisms use the energy to rearrange molecules in order to use it or store it for later use. Stored energy can be eaten by other organisms who may themselves be eaten. When an organism harvests energy, it can use the energy to conduct cellular processes, keeping itself organized, and it can use the energy for growth and reproduction. We probably wouldn't study biology if organisms and their abiotic environment didn't interact with one another. Because energy flows through biological systems, molecules are constantly being rearranged, and are constantly reacting with one another. It's the details of these interactions and how the systems that self-regulate them makes biology interesting. Now, often when we think of how diverse life is on Earth, we often think of things which are the most familiar. For example, if you think of a list of diverse organisms, you might think of an elephant, a whale, an ant, a tree, grass, an earthworm, and a mushroom. And we often leave out all of the organisms without nuclei, or prokaryotic organisms, even though they are collectively much more diverse than all of the eukaryotes, which only occupy this small portion of the tree of life. The process of evolution is responsible for generating all the diversity despite the common origin of all life. The term evolution basically refers to the simple idea that populations of organisms change over time. Although we don't study evolution explicitly in this course, you should be aware that natural selection is only one of several processes that cause evolutionary change. Many characteristics, including behaviors, that some organisms possess can be passed down from parents to offspring, and are more likely to be passed down if that characteristic or adaptation helps the individual to survive and reproduce. Individuals without that adaptation are less likely to pass on their genes, and so over time the population changes and the adaptation becomes more common. You'll watch a video about how fur color is an adaptation to different soil types. Notice that when we talk about evolutionary change, we don't talk about the change in an individual. Although individuals do change over their lives, that change is growth and aging, not evolutionary change. Evolutionary change specifically refers to the changes that happen in the makeup of a population or a group of individuals. Another thing to note is that individuals survive and reproduce, or not, due to their own tendency to do so. They reproduce to pass on their own genes, and they never do so for the good of the species. In general, each individual doesn't know or care that their species may be going extinct. Their drive to reproduce is a selfish act to maximize their own fitness or reproduction, not to save the species. So evolution explains how diverse life is, but it also explains that organisms have similarities, and these similarities are due to the fact that we're all related. As relatives, we share the same characteristics, such as all having our information coded as DNA, and the DNA working in pretty much the same way throughout the tree of life. We share many cellular processes with our relatives, the bacteria and archaea, distant though they may be. Those similarities are easier to see when we look at closer relatives. We have some of the same genes as fruit flies, cows, lemurs, and chimpanzees. The closer the relative, the more similarities we have. And this is true whether we look at morphological features like arms and fingers, or whether we look at molecular features such as the presence of particular mutations in our genes. We'll learn more about how mutations affect individuals later in the course, and in later courses you'll learn about how these mutations affect populations and are the source of the raw material that evolution works on.